Okay. Perfect. Thank you. Thanks again, Alex, for accepting our invitation. So let me very quickly go for a very brief intro. Dr. Alex Casson is an associate professor in the Materials, Devices, and System Division of the Department of Electrical and Electronic Engineering at the University of Manchester. His research focuses on non-invasive bioelectronic interfaces, the design and application of wearable sensors and skin conformal flexible sensors for human body monitoring and data analysis. The way that's actually, so this kind of collaboration with Alex started was back to, if I'm not wrong, 2020, that we applied for a seed grant, seed joint grant between University of Toronto and University of Manchester for the use of these kind of electrodes that Alex created in his lab to use with for people or patients with Parkinson's disease. And Alex now is in Toronto, and during his visit, we are going also to visit Dr. Chen's lab. This is also kind of part of this collaboration. Without any further ado, Alex, please go ahead.
Okay, thank you. So thank you very much for that introduction and for all coming. It's great to speak to you all today. Right, I've got a kind of a table of content for my presentation, and there's a lot of stuff on here. I've got a lot of different slides, and that's kind of deliberate. I'm not going to go into all of the things that are in this slide deck in detail. What I wanted to do was kind of touch on quite a few different areas, because my lab is working on quite a few different areas. And something I would really like to do with my presentations, I don't know how realistic it is to actually do, is to do a little bit of kind of choose your own presentation. So if there are particular areas inside here that you'd like me to spend a bit more time on or a bit less, just put it in the chat and I can focus down on that. Otherwise, I'm going to go through, I'd say, some of these slides really very quickly. Apart from the kind of background and big picture, because I think everybody has to sit through that. So we start off with this kind of background. I always like to put acknowledgements at the start of these presentations, because the work which I'm doing in my lab is very collaborative, both with international collaborators like Milad here, and with people across the UK. I think Liam is one of my PhD students, and you can see him there, hopefully. And hopefully you're in one of these pictures. Yeah, Liam's in our visit to the crystal maze that we have here. But in terms of the work that I'm presenting, it's very collaborative. You could easily get to 100 people working across the different aspects of these projects. And if you're including our long COVID work, which I'm not really going to discuss in this presentation, it's probably closer to 150 at least. Just for those of you who aren't familiar with Manchester, we're one of the largest universities in the UK with about 40,000 students. Well, I think that's relatively run-of-the-mill for kind of North American standards. Most people, I think, have heard of Manchester because they're either kind of fans of the cricket or fans of the football. Do we have any United fans with us today? Normally there's at least somebody who's just a United fan, but I go through say
only Chelsea here.
Was that Chelsea?
Yeah, sorry.
Actually, I used to, when I lived in London, you could see into Stamford Bridge from the flat I was in. So Chelsea's okay.
All right.
I put my background about kind of non-invasive devices. So it's all about interfacing with people and connecting between kind of the person, the physiology on the left-hand side here, and the electronic world on the right-hand side. And of course, that's a bi-directional thing, so going up and then also coming back down. And today we would call those sort of things kind of wearable devices. If you go back kind of more than 15 years ago to when I started in this sort of area, we didn't have these sort of low power portable electronics that we have today. And my PhD was very much doing microelectronic circuit design, doing the electronics to go into portable devices. If you go to my lab today, then we've got activity across that complete spectrum. So doing not just the kind of electronics to go into a device, but the kind of flexible interfaces for connecting to the body with not a rigid device like we might use today. Body phantoms in order for kind of testing our devices. We're not a kind of a big data lab. We don't do kind of data mining type stuff, but we do a lot of machine learning, artificial intelligence, normally in a constrained type way. So by constrained, I mean it might be power constrained, because you've got to operate on a limited battery life in a portable device. Or I think actually more interesting is when we're time constrained. So if you're wanting to make closed loop devices, and I'll speak about that a lot today, we often don't have time to send the data from you to a data center and back again. And then, as I say, we've got a lot of work about actual kind of closed loop devices, different ways of interfacing with people. And yeah, there's a couple of Man City fans in there. Super. So you can see Man City from the building that we sit in Manchester. And I say this is very multidisciplinary work, and certainly in the UK, we've been trying to build up just kind of cross-disciplinary connections for doing this. So I sit principally in an engineering school. I am an electronic engineer by background. That's what my degree is in. My office these days is in what's called the Henry Royce Institute. So this is the UK's national facility for materials research. And because that gives us access to facilities and kit and stuff, which isn't necessarily available at the single university level. But then I'm also a fellow of our Maxing Institute for Data Science. So really trying to leverage the national infrastructure that we have for connecting between the kind of flexible electronics for interfacing with a person with the kind of data science for doing stuff with all of that information that we can get from somebody. And then for reasons, I'm not sure what reasons, I have an appointment in a medical school, which is in Leeds rather than in Manchester. So that's about an hour away from us. And then we're currently doing an actual hospital-based appointment. So really trying to interact on a day-to-day basis with all of those different disciplines. And then I think Milad touched on this. The first time I came to Toronto was actually back in 2018, which is when we were setting up our collaboration schemes. So 2020 is when we applied for funding under that. And last year, the University of Manchester, the University of Toronto launched a joint translational centre for digital health. And they're what is funding us right now. And I think actually that the next call for funding actually went out last week. So if you are interested in doing transatlantic collaborations, there should be some funding available, I believe, right now for doing those sort of things. So if you go back to my kind of cross section of going between you and the cloud and back again, I would kind of break that down into kind of 4 different areas. So one is the kind of up route, so the sensing and getting data from you up to the cloud. One which is about the kind of signal processing, the machine learning, doing stuff with that. One is the down route, the actuation, the feedback, the treatment route. And I think we're going to talk about a lot about that in today's presentation, because I think that's where a lot of the kind of frontiers are, certainly in terms of, say, brain interfacing. And then lastly, in terms of applications. So that's really kind of using devices in the NHS in the UK context. A lot of that work today has been taken over by, as I said, long COVID activities, which I'm not really going to go into detail on. Now, within that big spectrum, Okay, we've got a debate about football going on. Within that spectrum of activities, I think undoubtedly my lab is best known for its work on non-invasive brain interfacing. By that, for recording, I mean the EEG. So that's measuring the electrical activities within the head with kind of small electrodes on the outside. I imagine most people on the call are familiar with that. And if I can direct you in the direction of my own resources, we've got kind of two kind of position kind of papers on kind of wearable EEG, kind of doing these sort of things out of the lab. And I think we've got one book chapter, one review article. I think they really do a good job of going from nothing to getting you up to speed very quickly. And then, because that's the kind of up route, that's the recording route. The going back is about brain stimulation, and we've explored a number of different ways of doing that recently. So in the setup here, this is an electrical stimulation electrode switch for putting electrical current into the head. And then the kind of roadmap in terms of doing brain interfacing, I think it is along the lines of what we have on the screen here. And this is probably kind of similar to what a lot of different kind of groups have their eye on in this sort of space, which is to say we want to do closed loop, individualized neuromodulation. I think we're aware that there's a lot of targets that we could be kind of hitting with, I quite like the term electroceuticals. So rather than pharmaceuticals, doing some form of electrical interfacing with the nervous system. If you want to do that in the open loop, just kind of turning, say, a current on or turning off, that's relatively easy. There's some long-term challenges, but fundamentally you could probably do it. But it's extremely variable. The kind of effectiveness decreases over time, and I say you get very different performances between different people and between different people of the same person at different points in time. And so there's lots of work looking at how can we measure biomarkers, biosignals, principally I mean the EEG in this particular case, use that to then guide the stimulation that we want to do. And of course, ideally, we want to do that in a way that it can be portable, it can be robust to artifacts, it can be easy to use, and so on. My lab, we look principally at non-invasive ways. Of course, there are invasive equivalents of these sort of things, adaptive deep brain stimulation for Parkinson's being the obvious one. But we're interested in exploring the non-invasive stuff you could do as a precursor, potentially, before more invasive options. Right. Super. Right. So within that, I am just going to go through a few different topics towards enabling that that sort of kind of personalized neuromodulation. Now, of course, you can personalize in at least two different ways. So one way is through kind of data-driven personalization. So that would be measuring your brain state right now and say matching the stimulation to your individual alpha frequency, as it were, rather than just stimulating at 10 Hertz. But you can also personalize through the physical form factor. And by that, I principally mean the kind of electrodes that we use for doing the recording. So I've already given a bit of an introduction to EEG. I think most people here are familiar with what that is. The key challenge in terms of EEG instrumentation is, of course, getting a good contact with the scalp, despite the presence of hair. Historically, we've used wet electrodes. We've used a conductive gel with some silver fluoride electrodes in order to get a good low contact impedance with the scalp. In more recent times, people have created fingered electrodes. So kind of electrodes with little prongs that can come down and kind of push the hair apart in order to get a contact with the scalp without needing a conductive gel. You can buy those sort of electrodes. Typically, they're made out of gold or silver chloride. But what they are is not personalized. You get a single kind of shape, size of electrode, despite all of the different types of hair, the different shapes of head, and so on. And of course, there's a big direction in EEG at the moment about inclusive EEG, having electrodes that aren't just 1 centimeter discs, electrodes that can be kind of tailored so they get the most appropriate fit for all different people. So in terms of, say, a fingered electrode, you can see there's quite a number of different degrees of freedom. What we looked at was, could we 3D print these shapes? Because if we 3D print them, we can make them on a on-demand basis, and to tailor the size, the shape, and so on that we get for different people. And the kind of short answer to that is, yes, we can. So this is an example of our current electrodes, which are in a TPU 3D printing filament, and we then coat them in a medical grade silver chloride. So this is the same sensing material, which is used in kind of off-the-shelf electrodes today. But we've got a relatively thin layer of it such that it can flex, and we can get kind of personalized, flexible electrodes for hopefully getting a good level of comfort. And we've got some example signals that we have here. So this is just an alpha burst type thing. So when somebody closes their eyes, you see about 10 hertz activity at the back of the head. And for all of our different electrode types, we can see those sort of alpha bursts being recorded there. We're operating these ones as dry electrodes, but of course we could add conductive gel if we wanted to turn them back towards a more conventional electrode case. When we use them in dry mode, we do find we pick up more noise from the contact impedance, and that's just a kind of a materials interfacing type thing. I think for most applications, that's what we're interested in, what our current state of the art is. But we have also made directly conductive ones now. So with the silver chloride based ones, what we do is we print an electrode. It takes you 10 minutes, whatever, and then we coat it in silver chloride. And of course, that coating process is what dominates how long it takes. It's not really an on-demand service there. Now we're using a carbon-loaded TPU, which is conductive as well as being flexible. And it can record the EEG without needing that coating process. So it's much cheaper and it's much quicker. We've got an example here of a steady state visual evoked response. So, oh no, sorry. No, this is going to be an alpha test again. So this is again somebody closing their eyes, and we're seeing the eyes closed. We're seeing a peak at about 10 hertz, and we see that in all of the different electrode types. These work. What they do have, of course, is a very high contact impedance to the body. So they are very, very sensitive to motion artifacts, much more so than the traditional silver chloride ones are. So you kind of get benefits in terms of cost, in terms of speed, but more artifacts coming into the collected signal. One of the key bits of infrastructure that we put into place for kind of making all of our electrodes then was looking at making phantom heads. So when we're doing our novel electrodes, of course, we can put them on a person potentially, but we don't know what they're meant to record, because we don't know what somebody's brain is doing at a particular point in time. We can give them an evoked response to record, get them to close their eyes, clench their jaws, whatever, but you still don't know what the actual signal is. And so we created a phantom head for doing this. So we've got some internal electrodes that let us play out a signal so that we know what it is to record. And I think actually quite a few labs have their own internal phantom head, but very few labs had kind of quantified or put into perspective, kind of had published accuracy on how good their phantom heads are meant to be. So our head is made from a ballistics grade gelatin. We 3D print a mold that looks like a head for it to go in, and then we can add in various impurities, principally salt, to the gelatin in order to control the properties, trade-off between the electrical accuracy, the mechanical accuracy, and so on. And as I said, we've got a number of different configurations. With just two on screen here, but we can have a look at the accuracy of those phantoms, and also then how they compare between what we would expect from an electrical model from the literature, but also just from measuring biological tissue. This is a porcine skin sample in this particular case, and there's a bit of variability in all of them, but broadly it provides a good model of the electrical accuracy of collecting the EEG, and we use that a lot for doing controlled testing. So often we don't make a complete head shape, it's a bit overkill. We have a kind of a test tube, and then we can control the pressure. We can look, we know what the comfortable pressure range is for putting electrode against the head is. And so we can look at how the noise scales with contact force, how the things like that. Something we found, which we weren't quite expecting, was we know that the kind of contact impedance changes as you put electrodes against the head more. Essentially, if you're willing to push something in till it hurts, you get a better contact. That's probably not surprising. What we also found was that the group delay of the electrode changes. So when you pass a signal through an electrode, it gets distorted slightly by what's called the group delay. Not all frequencies are passed through in the at the same speed. And so some of the peaks get moved about just a little bit. And so we knew that things like noise scaled with contact area. But actually we find that the group delay of our electrodes scaled with contact area as well. And we were able to find this out because we could do that sort of controlled testing. It's in the kind of level of microseconds. So for most experiments, it probably doesn't matter. But certainly for some of the configurations, you are getting up to 500 microseconds or so, which we might well start to matter for some people. Now, that's the kind of 3D printing of electrodes. What we've also done a lot of work on is 2D printing of electrodes. And this is essentially our work towards some of these, what I would call conformal type devices. So this kind of temperature 2 type structure, this isn't one of mine, this is coming from John Rogers at Northwestern, but essentially looking at the kind of next generation sensing platform. So not conventional electronics that's been miniaturized so that it fits onto your forehead, onto the wrist, whatever, but something that can actually be like an artificial skin layer for providing that kind of an artificial interface to the body. In my lab, we've done that a couple of different ways. So one way is by inkjet printing. And so in many ways, this is very similar to an inkjet printer that you might have, but now we're using conductive inks. And so again, this is it's either a silver or silver chloride. I said I'd check that. This is a silver one, onto a temperature 2 based substrate. And of course, the limitation of this very kind of 2D structure, unlike the 3D ones that we just saw, is that they can't penetrate through the hair. But of course, there's a lot of work out there looking at behind the ear, EEG, the kind of EEG you might be able to collect from a standard hearing aid location. And again, we've got an example here showing it collecting a steady state visual evoked response, in this case at 11 Hertz. Much of our work, rather than using inkjet printing, is using screen printing. So the benefit of inkjet is that it's a digital method, so you can kind of personalize, you can change the design each and every time you print it. The negative is that it's very hard to scale up. So it's very easy if you want to make a few of them, much more difficult if you want to make lots and lots. Screen printing, on the other hand, is relatively easy to scale up, but you have a fixed mask. So your kind of shape is predefined. And so this sort of printing is what Liam, who's with me in Toronto, is working on, and he's put them onto a wide number of different substrates. I think 25 micron thick PEN is probably it's probably not the default choice these days, actually.
I think two-stage, but yeah, there's very, very flexible things. I think the temperature 2 goes down to 500 nanometers, so it's very, very fun.
Yeah. And so there's a number of different ways in which we can make them. This is taking a look at them attached to mouse skin. And you can see kind of different levels of kind of conformality, different amounts to which we're kind of connecting with the skin there. And Liam's done work looking at the kind of impedance of the connection. So a bit like with the phantom head, but now actually measuring the contact impedance to the mouse skin layer rather than a materials-based phantom. And then also doing histological slicing, so we can take a look at what does the contact actually look like. These examples are for heart sensing rather than for brain sensing. I wouldn't worry too much about that. It's extremely similar, but in different places. And we see some very nice signals to high signal to noise quality recordings there. And again, this is just the modeling of the contact impedance. You can see the kind of capacitive connection to the body that we're getting there. And then just some examples of different contacts that we're getting after the slicing process. So it's quite hard to keep the electrode attached while you're slicing it up to put it onto a slide. But we're getting some insights into, let's say, the trade-off between different materials, different substrates, different skin contact qualities, and so on. Now, one of the key things we're looking at in terms of screen printing, our interest is that we want to do scalability. And of course, we don't want to do just the electrode. We ideally want to do the complete system. And so this is our complete system. This particular one is sized actually for heart sensing rather than for brain sensing. That's just because of the particular project that is we're doing there. But this is all screen printed. So we're using silver tracks, UV curable dielectrics, and then isotropic and anisotropic conductive pastes for mounting small components onto flexible substrates. And kind of making these sort of flexible electronics forms a big part of what my lab is looking at. And really we're looking at scale-up compatibility, because with screen printing, you can make them on what's called a roll-to-roll process. So it's not like a printer where you put in an A4 bit of paper, get an A4 bit out. You can get 110 meters on a roll, and you just run it through. And so this is KCN, one of my postdocs. I took this on Thursday, just before we came out, and this came to us in the post just the week, just last week. And so the idea is that this is then our base platform. We've got some flexible electronics that we can use, I say for this case, for heart sensing. But then we can start to We know that we can scale up and manufacture at scale. It's not a small prototype, we can only give you one of them kind of thing. And now we can start to play about. We can swap things in, swap things out. So we're looking to add, say, sweat sensing onto this platform and scale it in exactly the same way. We've got an example in the middle here where we swapped out the plastic substrate for paper. This is literally a bit of paper we got from a printer. We swapped out the silver to look at carbon-based inks. And the idea is we can start to explore some of the sustainability aspects, get rid of kind of electronic waste, essentially. And of course, that would form a key part of a complete system. I mean, I'm not going to go into lots of details in this presentation because there's not lots of time. But I do a lot of work about wearable devices in general. So this would be our next generation flexible electronic wearable. With NHS partners in the clinic, we're doing a lot of work giving people different devices. And so we make kind of electronics for putting into those devices. These would be relatively standard electronics, kind of rigid FR4 type ones for different functionalities. Right, and I will skip over this one, just in the interest of time, and go on instead to just talking a little bit about neuromodulation. So we've spoken about kind of personalizing the kind of closed loop non-invasive brain interface that we have from the kind of physical point of view. From the data-driven point of view, I think that really comes down to the closed loop, changing the stimulation that we give to people based upon the data that we're collecting in real time. Now, there's lots of different ways of providing neuromodulation. The ones that we've looked at in my lab are kind of on the left-hand side in red here. And we've looked, a lot of this work has been done for kind of chronic pain-based applications, but we've done some work in things like Alzheimer's models, in epilepsy models, and similar. Now, for those of you working on neuromodulation, I think you'll be familiar with a lot of these negatives. There's a lot of debate in the field. I think I would summarize it as a lot of the results are just variable. There's lots of studies that don't replicate or find slightly different things. And some of the causes of that are experimental, but I think a lot of them are technological as well. I think there's scope for much better technologies for doing our neuromodulation. I started off in this space by looking at electrical stimulation, principally AC, so transcranial alternating current stimulation, where you put in you inject a sine wave of current. And of course, the key challenge there is that all you see is the artifact. So obviously with the EEG, you're measuring voltage, you're measuring the electrical properties, and when you inject current, an electrical thing, that's what you see. And so even today in the literature, the vast majority of studies that you'll see are comparing EEG from before the stimulation is done to the EEG after the stimulation is done. And there's just a big kind of section in the middle where there's no info, and you kind of hope that the two ends connect together. So in my lab, we've made then a number of different platforms for doing closed loop neuromodulation. The two on the left are kind of lab-based systems. The two on the right are mobile phone-based systems. So they are kind of portable. You could take them. We've got a Bluetooth streaming EEG headset. And so we've got a study on neurofeedback at the moment, which people are doing in their home environments using a provided mobile phone. And then, Mila, let me know when I've been speaking too long. Otherwise, I'll just whiz through some of these things a little bit too, a little bit. So, how long do you want me to speak for, my lad?
Well, I think officially you have time until 11. We, at the same time, appreciate for question and answer. So what I suggest is, if anyone has a question, please feel free to ask Alex now or even during his next slides. Otherwise, I think we can continue.
Yeah. I'll keep going. I won't say too long, right? So sleep engineering is this kind of concept of manipulating your brain while you're asleep. And there's a relatively pivotal investigation by someone called Hongi about 10 years ago now, which showed while you're asleep, there's a feature known as slow oscillation. We've known about that for a long time. And if you play an auditory sound at the peak of a slow oscillation, you can trigger another slow oscillation. And so you've got a kind of a time constrained situation. You've got typically 50 milliseconds or so between this trough, that's when you know a slow oscillation is occurring. You have about 50 milliseconds to make a decision. That's why you've got kind of time constrained signal processing going on. But then to play a sound, and potentially to make trains of slow oscillations. And of course, we know that slow oscillations are very correlated with your memory consolidation performance. Essentially, kind of young, healthy people have lots of slow oscillations. You have fewer as you age. As you move into mild cognitive impairments, you have essentially none. And so, there's potentials, and I think it is early stage stuff for looking at kind of non-pharmacological routes for helping with memory performance, but also just helping explore what the brain is doing. So we made a platform for doing that, and you can see an example here where you can see very nice trains of slower oscillations being formed based upon the stimulation that we gave them. The interface that we did for this was very deliberately in MATLAB because we can tailor the stimulation that we give very quickly and easily. So we can give them 10 clicks or 2 clicks or a longer delay or a shorter delay very quickly and easily. And we've got lots of results for doing sleep engineering. We started off by doing a placebo experiment. So just kind of telling people that they were going to receive this sort of stimulation, and we would hope it would change their behavioral performance. And we didn't see any placebo effect. We did possibly see a slight nocebo effect. So kind of people who weren't expecting it to work, it didn't work. Sort of a thing. In terms of the behaviorals, I think we've still got more work to do here. I think what we found, like a lot of other labs, is that we do find changes in memory performance, but they're very, very specific. It's not kind of across the board. You change the task slightly and the effect goes away. But it does seem to be relatively robust for certain types of tasks. And we've got a little bit of fMRI evidence to help support such a view, doing some longer term studies, kind of multiple nights of stimulation. In terms of smartphone based, so when we speak to users, what we often find is they're expecting the interface to be app based, kind of phone based. That's what the embodiment that they would fundamentally like to see. So that's driven a lot of the sort of work that we've done in this sort of space, because if we're constrained to a mobile phone form factor, essentially we're constrained to visual and auditory stimulation. And so we've made apps for doing both of those. So in the visual case, it is just flashing the screen at the stimulation frequency that we would like to provide. In the audio case, it's one of providing binaural beats. So typically we do 400 hertz to one ear and maybe 410 hertz to the other ear, and you can get a modulation frequency of about 10 hertz. That's all gone through kind of the co-design process, just helping users to guide what they would like kind of interventions to look like. And from a technical point of view, it works. We've got some results in there. And we've got a clinician who is currently doing experiments with people using this sort of stimulation at night before people go to bed and having self-reported pain scores. I won't name names. Steve's a great clinician, maybe not so good at making graphs. I think other students I would have asked to remake this, but fundamentally, you know, these lines are doing what we would want for a non-invasive, non-pharmacological, easy to use, portable intervention. They are looking at decreasing self-reported pain. And then, as I say, one of the key challenges in audio EEG, as I'm sure you know, is about motion artifact robustness, and we've done work for our portable platform to be robust. For both of those stimulations that we've seen, they're very kind of passive on the person. So you sit there essentially, and something happens to you. Neurofeedback is the more general. You're being in the loop. You're asked to do something to kind of change how you're in the loop. You're consciously thinking about stuff. So typically people are given a dial on a screen and are asked to put the dial in the green zone rather than the red zone. And they might not be given any more info than that. It's for people to self-learn how to manipulate the feedback. So it's a different route for doing the modulation. And again, we've got a portable platform for doing this that we're currently trialing. For the in-lab tests of that, so we don't have any results from the at-home study yet, but again, they're broadly showing what we would want. So if we ask people to down-regulate their alpha activity, then we see no changes. If we ask them to up-regulate, so between these two cases, we're just literally changing the dial. Is it going that way or is it going the other way? And we can see decreases in self-reported pain scores the more neurofeedback people are doing. And then lastly, and I will just whiz through this very quickly. We've seen this before for the electrical sort of stimulation. The big challenge is artifacts. We can't start to close the loop. We can't start to do the personalization until we can see the brain activity that's going on underneath all of this artifact. And so principally the way to get rid of that is in software algorithms for doing the removal. Now, of course, it turns out that making some software for doing artifact removal is pretty straightforward. We've got a number of algorithms that we can make relatively quickly. Convincing people that they work is much, much tougher. And of course, I think there's a bit of history here. You know, the people in the neuromodulation space are aware of things that haven't worked. They're aware that these things don't necessarily replicate and so on. And people are rightly skeptical. And of course, because when we're stimulating the brain, we can't see what the brain activity is. It's very, very difficult to verify the algorithms have worked. Because we don't know what we were meant to be recording. So we're trying to compare our clean signal to an unknown signal. And yeah, just to put the kind of problem in context, this is a standard EEG signal. This is what the artifact looks like once we're doing the electrical stimulation. And the signal's noise ratio is about -40. And it's rare you see other biomedical engineering problems with such a low signal to noise ratio. There are one or two out there, but it really is a challenging artifact removal case. Let's go over those. So we've got, as I say, we've got a couple of different algorithms for doing this. Let me just mention some of the verification stages that we made. SMA would be our default one. There's a toolbox online you can download that implements it. But the first thing we did actually was use our phantom head. Now we've already mentioned this, and actually we first made our phantom head not for making electrodes, for testing electrodes, but for testing our tax artifact removal algorithms. So we don't just have internal electrodes playing out EEG. We do tax to the phantom head at the same time, and of course that gives us a known signal to compare against, to know have we recovered the correct thing. I think we do find that the artifact removal problem on a phantom head is a bit easier than on a real head, but on the phantom head it performs very well. You can see the correct signal being recorded. And again, because it's controlled, we can start to see what the non-ideal effects of the algorithm are. So pretty much all artifact removals involve some form of memory, taking some form of average over a few samples at least. And that means you get artifacts when you turn the stimulation on, when you turn the stimulation off, because you've got a kind of discontinuity within that memory period. And we can tailor the algorithm depending on how do we want the artifact at the end, do we want the artifact at the start. And ultimately, this will limit how quickly you can close the loop on your stimulation. Done various on-person tests. So again, you can see various alpha bursts here. We find that the 40 Hertz case, just with higher frequency, particularly for the adaptive filtering, is the worst case. I think probably we didn't sample the signal quickly enough. So our EEG is sampled at 500 Hertz. And we're stimulating at 40 Hertz. I think we just need to oversample a bit more, really. Certainly, for the big morphological features, you're probably not interested in seeing an eye blink, but they are recovered quite nicely. We can see the eye blinks during stimulation. Again, we can see kind of 10 Hertz when you close your eyes, alpha activity occurring. The descriptive statistics are exactly the same. We can see evoked responses. So here we gave somebody a face recognition task, so we can see N170s occurring when they see that it is a face. So all of these are real data. They've had that big tax artifact removed. And then lastly, what we also looked at was machine learning verification. And so what we did here was give people two different tasks to do. So in some ways, these are quite similar tasks. They're both looking at different bits of working memory. So one is asking you to recognize when you last saw a particular symbol. That's an N-back task. And then a backwards digit recall task is asking you to think backwards about the different numbers that you see. And we did this at both with tax and without tax. And so what we find is that if we just look at, did somebody do, when somebody's doing the task with stimulation or not with stimulation, can we identify those different periods? And the answer is that the machine learning can. And of course, that's what we would expect if there were lots of residual artifacts in our cleaned trace. Right. So in the stimulation case, task plus stim, if we've cleaned it, but it's not very good, the data will look very different to the task cases, and it'd be very easy to get a good separation between those two different tasks. But then, of course, what we did further was the 6-class classification problem. So we didn't just look at, can we tell, are you doing the task or not? We looked at, can we tell which class it is you're doing? And the answer is, it's not perfect, but yes, we can. And so either this means after we've done our artifact removal, either we've got residual artifacts that are specific to which task you're doing, enabling you to separate them, Or, and we would hope for this, but in this case, we've got EEG components, which are different to the two different tasks. And we think that this adds quite a nice, certainly use of, we're really spanning that spectrum from phantom head to data science and using that to do verification that we are getting what it is we want. Right, I've touched on a lot of different bits there very quickly. Very happy to kind of accept any questions. But as I said, hopefully you can see when we start to put together some of our flexible electronics, some of our personalized electronics, some of our data-driven personalization, we can get things which are very different to what we have right now.
Great. Thank you so much, Alex. That's a very great talk. We are going to start questions. Paul, please.
Yeah, can you hear me?
Yep.
Yes.
Yeah. Thank you very much, Professor Casson. Anyway, it was interesting and all are stimulating to me. It looks that we are in the field. Okay, so my question is regarding the phantom head. So when you created the phantom head, did you consider the skin, bone, and the brain tissue, the parameters or characteristics?
So the short answer to that is no. This is a single layer phantom. When you look in the literature, there's a bit of a split. There are people doing 3-layer phantoms exactly as you suggested there, typically filling them in with gelatin like we did between the layers, or there's people doing homogeneous single layer ones. And we've always gone that second route. I think partly because it's a bit just physically easier to do, partly because we've got good results doing that. When you look at the kind of electrical accuracy, although it is just one structure, we've been very happy with the accuracy that we get. But also, I think for me, what we've been trying to do and not really succeeding yet isn't so much more modeling inside the head. I think we're happy with that. It's trying to get hair onto the thing. And that's the much more kind of pressing need.
Okay, thank you.
Maritza?
Yes. So thank you, Alex. So I have a couple of questions. Probably they're a little bit technical because I'm not from the field here. The first one is just wondering about your electrodes. You show that they're extremely sensitive to motion detection, and I assume deformation, right? So how is this deformation play in the detection of your signal? I mean, I assume, right, that they are generally small. So probably in other contexts, I would say that you can model these electrodes as point contacts on your scalp. But the signal that you're recording here is extremely small, right? So how the actual deformation of these electrodes on the point of the contact is playing on the signal to noise ratio in your data.
Yeah. So firstly, what I would say is it's principally the directly conductive electrodes that are very sensitive to motion. The silver chloride ones would be the same as it were as current electrodes. It's just, as they may take very longer to make. Now, in terms of the kind of impact, there's a couple of different ways of thinking about that essentially. So when you're kind of, if you imagine your fingers coming through the hair, right? Well, you've got 2 factors. So one is you've got the contact area, and then the other is you've got the contact force. And of course, those 2 interact if your electrode is going to spread and push them and kind of push the legs apart. Now, in general, you will get better performance if you've got more contact area and if you push the electrodes in harder. Both of them are roughly linear. So if you push in twice as hard, you can get kind of half the kind of contact impedance, as it were. And we've not.
So the actual deformation of the legs of the spider electrode, for example, is not a matter of concern here.
So the spider electrodes are size. So as you push them down more, they've got like they're not flat. They kind of curve out. So you'll get kind of ever increasing stuff. So in that case, it's like you're right. That one, it wouldn't be a linear. It's a linear relationship between say noise and contact area. That's not necessarily a linear relationship with how you size your electrode.
That I would assume. So what can you say instead of the characterization here is in terms of input conductance, right? So what about the output? So like, how do you transmit the signal to the skull here? Is it the same kind of characterization, or they go one by one, input conductance, output conductance, or should I, in terms of modeling, should I consider other factors?
Well, let me just not answer that question directly first. I think one of the key things, of course, that we've not done is to look at the dynamic nature of these things. So it's one thing having a static, I push this hard, the electrode spares this much, it touches by this much. In reality, when you're moving about, so particularly these things are under a cap, say, those things are going to be changing over time. And of course, that might present a different levels of contact quality at different points in time. And of course, when in terms of the generation of artifacts, what tends to matter isn't so much the impedance that you get, the absolute number, but the balance of impedances that we get across the head. So when we say typically we want a contact impedance of less than 10 kiloohms, that's not strictly true. What we want is all the electrodes to be matched to be within 10 kiloohms. And the easiest way of doing that is to have all of them as an absolute number be less than 10 kiloohms. We don't have the kit to do this. If you go to the Army Research Laboratory in the States, what they do is they take their phantom head and they put it on like a 9-degree motion thing, so they can simulate it walking. So you can look at these effects in a controlled way. Now, I'm not sure that answers your question.
No, it was, well, yeah, I guess you'd be, so that was fairly interesting. I was just speculating on other aspects, but we'll have the chance to actually chat later. So because I was speaking with Milad, probably join you. I see that you are in the other room, so
yeah,
so you're here. So I prefer to leave the stage to other people here. So yeah, thanks Milad. Thanks, Alex.
Liam's got his hand up.
Yeah, sorry, I have one question. You mentioned that there's a sound with a slow oscillation. What audio tune is that you have to play?
So Liam's question, just in case he didn't come up on the microphone, Liam's question was about what sound is it we play for the audio stimulation during sleep engineering. So most people use pink noise, so that's kind of 1/f noise of a 50 millisecond duration. I think that's relatively arbitrary. And it comes from just for having to pick something point of view. Actually, the graph I showed you, where we had that long train of ones, the paper for that hadn't come out yet. It's on its second round of reviews. It's nearly there. But that was actually looking at playing different sounds to different people. So there was the classical, say, pink noise sounds, but we also gave people vowel sounds. And I think the conclusion was that there's not a lot of difference between the different sounds. Just kind of 1/f noise is fine. I think what, of course, when you're playing recognizable noises to people, you're in a slightly different space. So there's a space I'm not in. So the interest over here, I can just look at it. It's known as total memory recall and memory replays. And so you can cue people with a particular sound while they're awake. And then you can replay some of those things while people are asleep. And we're in a slightly different space there, but people have done recognizable sounds as well.
Okay. Thank you.
Very great. I have a few questions, but I'll ask you later in our meeting. So there's no rush for me. Any other question from anyone? Okay, it looks you're fine for now. Great. Thank you again, Alex, for your great talk. And I think we can end today's session. And yeah, have a great day to everyone.
Thank you very much.
Yeah, thanks very much. Bye.
Bye.
Bye-bye.